Jai Hind everyone, good morning to all of you. So we are going to study the new some questions which is based on our immunology. Okay, so we will solve that question which we covered till now. We covered till now the antibodies. Okay, so the question number six, what it is asking, which of the following antibody is used by immune complex for complement activation? So if you remember, we read here only the complement activation is done by what? Will be done by IgM, and IgM is what? Pentameric in nature. Okay, with one J chain. You just remember it. Next question is, what is the next question? Question number 18. 14, na? Question number 14. Which of the following is the least abundant immunoglobulin class in the normal adult? So the least abundant immunoglobulin class in the normal DNA or normal adult is what? Okay, first, which is the most abundant? So IgG is the most abundant. After that, IgA. Its IgG percentage is 80% around. Okay, IgA percentage is around 15%, and IgM percentage is around 10%, and Ig is less than 1, and IgD is less than 0.5. So five antigen antibodies are there. Among these all four, IgE will be the what they ask, least abundant. Okay, Ig is the least abundant. Now question number 18. So the question number 18 is asking what? The match the following it is. Okay. So IgM, you can match it with the what? Pentameric complement activation. So A you match with the 4. So there is a two option, 2 and 4. So this will be your uh, moving forward options. Okay. So 2 and 4, you just go with this. So option B is matched with 2. So IgA can be naive B cell antigen receptor. Okay. So actually IgA it can be, and the here the third option what it is written, IgA is monomer, dimer, trimer, mucosal immunity. So it can be monomer IgA, dimer, but it cannot be trimer. You didn't see it as a trimer. And the third is that after that C IgD it will be helping in the B's, it will helping in the activation of the B cells. You remember? We already read about that. It will help in the activation of the B cell to produce antibodies. Okay. So C can act on the what is the okay, C can act as a two, and IgE is what? Monomeric, and it will bind to the mast cell or the basophil cell. Okay, so the answer will be four only, correct? Your answer is correct. So answer will be the four. And okay, question number 33. Question number 33 is what? Some question we skipped because 2 3 because we didn't read till now in the topic. Okay, cytokine release in response to virus infection is termed as the cytokines which is released from the virus infection, that is interleukin, interferon, monokines, lymphokines. That is what is your answer?
Wait a minute.
Okay, if you're watching on the video, you just increase your speed up at 4.5 or 1.75x, then you will see you'll get the good qualities.
Interferon.
Yes, interferon will be released from the virus infected cells. So, question number 34. Humanized murine antibody is one in which the heavy and light chain are from human. Heavy chain is from the human and light chain is from the mouse. Light chain is from the human and heavy chain is from the mouse. CDR from the mouse and the other region is from the human. So actually humanized murine antibody is like the it is uh, it is kind of human antibodies. Okay, so it is kind of human antibodies means it is not directly derived from the human. Actually, it will be derived as a monoclonal antibody from the other organism. Like suppose that example, the antibody developed for the cancer drugs, that are the monoclonal antibodies and that are found outside the human body. Okay, so you are taking a drug for the cancer, it is not forming inside our body as a antibody. So you made that type of drug to the different organism, so that drug, that antibody can target our cancer cells. So what is the important thing by which it will target our cancer cell? It will be target our cancer cell only if that CDR, the complementary determining region will be there. So complementary determining region from mouse and the other region are from the humans. So that could be the probable answer. Okay, so in this one the back frame is back the framework region is there in the variable region. There we will do the mutation in of the human antibody. Okay, and then will get the then we will get the antibodies. Okay, so that is produced, that is non-human antibodies, humanized murine antibody, in which heavy and light chain both are same, but the CDR is little different. Okay, that is produced outside. So the probable answer will be the four. Okay, next question is 33, 34, and then 47. Question number 47. The question number 47 is where it is. I think it is. So which antibody is involved in the type 1 hypersensitivity? So this topic we didn't studied, but you seen the type hypersensitivity is done by what? Which immunoglobulin is causing the hypersensitivity? Hmm?
IgE.
Yes. So at least if you, even though we didn't read about all the hypersensitivity one, two, three, you can go with that option. Okay. So IgE will be the type one hypersensitivity reaction. Okay. When we will read the hypersensitivity, we will see the other IgG, like how it is involved. Which of the following? Okay, next. Next question is 49. Question number 49. So, to combat the several viral infection, what kind of immune response should be elicited? Innate immune response, humoral response, cell-mediated immune response. Both cell-mediated and immune response, humoral immune response. What is your answer? Both. Question number four, like option number four. So
Uh,
humoral and cell mediated means what? Humoral means innate immunity. Everyone knows, the immunity which is we are having from the birth. So, but viral infection we cannot have protect with the birth immunity, innate immunity. So we require acquired immunity. Okay. So in acquired immunity, you will see the humoral response and the cell mediated response. So the humoral response antibody mediated, B cell mediated humoral response will be the B cell mediated. Why I'm saying B cell mediated? It will give the antibody, and antibody will be in the humoral, in the fluid, blood. So that is called humoral means fluid. So if antibody is there in the blood, then we will see humoral immunity. So antibody will be against the virus. Something. And cell mediated means like if the virus will infect any cell, so infected cell can produce so many virus. So we have to kill that infected cells also. So that infected cell will be killed by the cytotoxic T cells. Okay. So that cytotoxic T cells will provide you the cell mediated immune response. So both answer will be the both humoral and cell mediated immune response. And then 58. There is 58. Uh, Symptom of an allergy attack can be prevented by. How you can prevent the allergic attack? Hypersensitivity means what? It is something which will be response of the body will be hyper. If fever is coming in the normal way, it will increase. If you are sneezing or if you are coughing slowly for a normal response, it will be increased too much. The breathing can be affected too much. Okay, in the case of asthma and not. So you just tell me what, how you can prevent hypersensitivity. Want to answer in the?
what Pankaj?
What is your answer for this question number 58?
I don't
Huh? You don't
know.
know.
Like,
Okay, you can guess now, like not don't do guessing and all, but according to your knowledge, whatever you studied, according to that you can guess.
actually, not discussed that about the T cells, natural killing cells.
It's okay, but even though you the
This
options is there now, which you know. Okay, where this went, I don't know. Arey yaar. Okay, that was the last question. If you just go and read back your video, why this happened? Read back your video, and you can check the options. Okay, so the option was will be the blocking of the IgE. If the IgE will not bind with the basophil or the mast cell, then it will not produce any. What it will not produce? It will not produce any histamine, inflammatory inflammations, inflammatory molecules. So finally it will provide you the, it will block the immune response. So up to GAT B, they didn't ask too much of the difficult question. So, but if you like to read properly like this, then we can go with this the same manner. Okay. Like we are discussing little more, and today we are going to discuss about the cell immune cells. What we will discuss? Immune cells. So, beta, immune cells are what? They are, suppose this is a bone marrow. Okay, we'll see something. Okay, she is a baby, and her boom. There is one baby. Okay, so this baby is in the womb, and in this case of fetus, the all the immune cells, not exactly all immune cells, all the blood cells. Okay, you can see. So the blood, especially RBC, will form in liver. Okay, in baby. When it will become adult, in case of adult, the blood will form in the bone marrow. Blood will form in bone marrow. Got it? In the beginning of the embryonic development, the blood cells RBC will be formed from the yolk. Like in the beginning of embryo, RBC is formed from the yolk. Okay. So we are talking about what? Blood formation. Huh? So in adult, bone marrow will form. In embryo, yolk, and in fetus, you can see developing fetus. You can see from the liver. So these are all the organs. So what? Before going to this topic, I will tell you something important. Suppose this is a zygote. Okay, this is a zygote, and this zygote can give rise two type of origin. Like it can give embryonic cell, and it can give, or it can give extra embryonic cells. Okay, extra embryonic cell. So what is extra embryonic cells? Like if this is a inner cell mass, so the cells which is outside this, this is your trophoblast. Okay, the outer cell is what? Trophoblast, and the cells which you are seeing inside this is our inner cell mass. Okay, this is our inner cell mass. So this inner cell mass will give three layers. Okay, it will give triploblast, three layers it will give, and this trophoblast, what it will give? This is trophoblast, and this trophoblast can give the placenta and all. Okay, placenta it will provide. So from this inner cell mass trophoblast, all the body organ will form. Like bone will form. Okay, suppose from this only bone will form, and in the bone there will be some type of important cells. We'll talk about that. And there is one important cells. Got it? The story you are going to see properly now. Okay, what I am going to show you. The thing is that the zygote can give every cell of the body. So the potency of this zygote is called as a totipotency. What it is called? Totipotency. Okay. But embryonic cell or this Extra embryonic cell, it will give trophoblast only, and embryonic cell will give inner cell mass. Okay, so this is defined to some proper work. Okay, that is like the that type of cells can give different cells, but these are all what the first step, second step. Okay, so in the second step, what you'll see, beta, that will be the called as a pluripotent. Pluripotent. Okay, so pluripotent means what? The cells which can give other than the extra embryonic cells. Okay, it can form all cells, all cell of body except all cell of the body. Okay, so but not but not extra embryonic extra embryonic cell. Okay, but not extra embryonic cell. But if you see this type of cells, once the you will get the triploblast. After this triploblast, you got the this bone marrow. So whatever the stem cells will be there in this cell, this stem cells will give. This is a stem cell. All are called as a stem cells. Zygote under the inner blast, yeah, blastomers. All are stem cells. Okay, so stem cell having the dividing capacities, retaining always dividing capacity. So the stem cells which is in the bone marrow, this is our bone marrow stem cell. So the stem cell which is present in the bone marrow, that cells are called as a hematopoietic cells. What it is called? Hematopoietic hematopoietic cell. And these hematopoietic cells can give only blood cells. This can give what? Blood. So that is called as a what? That is called as a multipotency. Okay, multipotent. It is called what? Multipotent. Okay. So you've seen totipotency, the cells which can give rise any kind of the blood cells. Sorry, any kind of body cells. Either it can give extra embryonic or embryonic cells. That is called totipotency. You've seen pluripotent, like the cells which can give any kind of body cells, but not extra embryonic cells. That is called pluripotent. That this type of potency you can see in the mesometic tissue also in our plant. And multipotency means what? The cells which is determined to give or committed to give rise in one kind of cells. Okay, that is called multipotency. Okay, these are the three potency you have to learn. And now, what we will see, beta, we'll see important. Now we'll see the immune cells. Okay, so from hematopoietic cells, suppose this is my hematopoietic cells. Okay, and these hematopoietic cells can be of two type. Okay, it can give myeloid origin. It can give some cells which will called as a myeloid origin. It is happening because of some factors. Okay, some factor will bind to this hematopoietic cells, and the process we also what we are saying, this formation of blood, that is called formation of blood is called formation of formed element, you can say. Formed element means blood cell. Formation of blood cell is called what? Hematopoiesis. Sir, sir. Hematopoiesis. It will call hematopoiesis. Okay. The another is what? Lymphoid origin. So another is what? Lymphoid in origin, and this myeloid origin will give you two type of cells, two type of origin. Next, the myeloid cell, whatever you are seeing, this will give either one type of cell. I will tell you this, and this will be another type of cell. So this is called as a megakaryocyte origin or the granulocyte progenitor. Okay, granulocyte. Okay, I should write it properly here. Okay. Granulocyte or or megakaryocyte site progenitor. So this is also a progenitor cell. Okay, this is myeloid progenitor. So this is also my progenitor cell, and this progenitor cell will give you a two type of cells. Okay, it will give two type of cells, and these two type of cells are, one will be the RBC. Okay, and another will be what? Another will be the uh, okay. Another will be what? Megakaryocyte. So in RBC, no nucleus is there. Okay, no nucleus is there, and the count of the RBC is around how much? Around 5.5 million, and its lifespan is around, its lifespan is 120, and this is what? This is our megakaryocyte. This megakaryocyte is, once it will go to the blood, once it will go to the blood, then it will form platelets. Okay, it will form platelets. So this is just a fragment. Platelet is what? It is a fragment of megakaryocyte. Site. Okay. Now, other thing is that it has a big nucleus. It have a big nucleus. It has. Now we will come to the granulocyte. Okay. I should write properly now. I will tell you the example and properly separately we will discuss it. So the granulocyte is of four type. It can be, sorry, I written here megakaryocyte. This is macrophage progenitor. Sorry, this is macrophage or granulocyte progenitor. It will called, RBC is also called as erythrocyte. You remember? RBC is also called as erythrocyte. Now, if you see granulocyte, it is of four type. First is what? First is what was the first? Okay, neutrophil. Neutrophil. basophil, eosinophil, 4 mast cell, like this. Okay. In lymphoid origin, it will give three type of cells. Lymphoid origin will give three type of cells. That is B cell, T cell, and natural killer cell. So you just remember here from where you got the macrophage. I forget to write macrophage also. Okay. So the macrophage is coming from what? Macrophage will be coming from the monocytes. I didn't write monocytes. Monocyte. And this monocyte can give it can give macrophage, or either it can form a dendritic cell. The origin will be same. Okay. Like the from where monocyte is formed, from there only dendritic cell will form, and from there only and monocyte will be formed the macrophage only in the tissue when it will enter to the tissue. Some phases will be there from the for the particular organ. We will see it later. So B cell, T cell, natural killer cells, all you learned here. Now we have to discuss all the cells one by one. Okay. So these all cells are all the cells are if I will talk about the we are starting with the granulocyte. Okay. So then granulocyte, what are the examples? Neutrophil. If you will see the neutrophil, so its nucleus is like this. So its nucleus is multi-lobed. Okay. Nucleus is what? Multi-lobed. Nucleus will be multi-lobed. Its percentage in the WBC is around 60%. Okay. In WBC. So what its role it will have in the it is phagocytosis. Phagocytic. And its color will be it is both neutral and it is neutral in nature. Why it is called as a neutral in nature? Because it have the pink mark. It will be seen as a pinkish or bluish. It will not exactly bluish. Whatever the color you will mix with the pink and then the same color it will give. Okay. So that is this is actually the granules which is stained by some pigments. These are the what? Granules. Sorry, it moves somewhere. These are the what? Granules, which is colored by because of the some dye. So as you're playing Holi's colors, so the color will be of any kind, but they are neutrophil will are the playing with the color of blue or red color. So this is neutral and phagocytic in nature. Now we'll see next cell. Next cell is what?
Pankaj, you are stopping your screen.
Huh?
The
Okay.
screen is not visible.
Okay. Neutrophil. Then next you can write basophil. So basophil is what? Basophil will be the cells which will be having S-shaped nucleus. Which type of nucleus it have? It have the S-shaped nucleus. Okay, and we already learned about that something important. What is it? The lifespan of this neutrophil, you can write it here around 23 days. Okay, not 23 days. Sorry, around 7 days. Okay, 7 days. So basophil is here only, and now this will be having S-shaped nucleus, and it is whenever you will play, he will play with the holy colors. He likes the more basic color. B for buffalo, B for blue, and B for basophil. So the all words begins with the B. So if you'll see, if you'll give the methylene blue is the holy color for, not Holi, holy, the festival Holi. So it is a holy color for the basophil. So it will give you the blue color. The nucleus is already S-shaped, and once it will be binded with the immunoglobulin E, which if it will bind with antibody E, suppose it will bind with here. Whenever antibody E will bind here, this all the granules will start degranulation. Okay, this will start degranulation. So suppose this granule will come here and they will release all their chemical here. Okay, so this is what these chemicals are actually basic in nature. Okay, so the granules basically is what basic in nature. Basically is basic in nature, and these are what histamine, serotonin, prostaglandin, like this. Okay, so the function of histamine is what? Inflammation. Serotonin have that diverse function. You can say it is a feel good function. Okay, and prostaglandin is what? Smooth muscle contraction. Smooth muscle contraction. As whenever the you will have the injury or the damage on your skin, then it will contract the blood vessels, so the blood loss will be less. Okay, serotonin will give you the feel good factor kind of thing. Means what? Whenever you will get the injury, though you will be very hyperactive, so it will try to balance that hyperactivity, and like this histamine will produce the inflammation. So once the inflammation will be done, so more uh, WBC can come to the site of the injury. Okay, now what it is next is what our next is eosinophil. Okay, eosinophil, and this is what eosinophil is bilobe nucleus cell. This is your eosinophil. Okay, so this eosinophil is bilobe nucleus. And which color it like, beta? It will like eosinophil means it will usually stand with the eosin acid, eosinic acid, and it will give the red color. Okay, or we can say red or pink, kind of pinkish. Okay, okay, this will give what? Some pink color. This is what? eosinophil. So it have one pink color on his nose also. So whenever allergic reaction, it will produce the allergic reaction also, and it will very important for the first is what this is the bilobe acidic in nature. How it is acidic nature. So they all have the granule, and the granule is colored in form. Okay, what is the percentage of it? It will be the
With the person.
percentage 2 to 3%. Okay, what is the percentage? 2 to 3%. And the important thing is it will help in anti-parasitic. Okay, anti-parasitic. Along with this, it will give response, give allergic response. Okay, it will also give allergic response. It will produce what? Some H2O2 peroxides or the reactive oxygens. Okay, you can write O2 minus. It will produce O2 minus reactive oxygens. It will produce reactive oxygen. So so pathogen cannot survive. Pathogen can't survive. Okay. So cationic peptides it will give like this. So all these things it will be given, and finally the pathogen will be evade. So if it is written here, the anti-parasitic means parasite are so many types. So you just mention it here in your notebook. Helminths it will product from the helminths and protozoa. Okay, I have the space for protozoa like protozoa. Okay. Now the percentage of the basophil is less than percentage of the basophil is less than 1% basophil. Percentage of now we'll see what we'll see the monocyte. We'll see the monocyte. Okay. So the monocyte is what? Monocyte is having a one nucleus. It doesn't have any different special kind of shape. And this monocyte having granule, having having none colored granules. So it is called as a granular. So this is called what? This is called a granular WBC. Okay. This is called as a granular WBC because the granules are there, but it is not colored. Okay. So having no colored granules, and the function of its function is actually it will help in the forming of macrophages. So we will go directly to that point. Okay. So the suppose this is our blood vessels. Okay. This is our blood vessels. In this blood vessels, the monocyte was moving. Okay. And this is a site of injury. What it is? Suppose this is my skin, and here the injury is happen. Okay. So here the injury happened. So or here the bacteria came. Okay. Here the bacteria came. So many bacteria is coming here, invading, and because of this, the macrophage, this monocyte will come inside. this this monocyte will come inside this, and there will be some process. This monocyte will be squeezed like this, completely squeeze. It will squeeze like this, and finally it will come inside the cell. When it will come here, then he will become aggressive. Okay. So it will become what? Macrophage. Along with this, it can form, the monocyte can form another type of cell which is like this. So that is dendritic cells. And these are all antigen presenting cells. Okay. Macrophage and dendritic cells are both, plus both are antigen presenting cell. These are all antigen presenting cells. Now these are the material. These monocytes are what? Blood streaming forms. Okay, some macrophage will be the uh, resident of the tissue. Okay, so there will be some tissue resident macrophage, and this tissue resident macrophage are like, it is, if it is in liver cell, it will called Kupffer cell. If it is in the brain, then it is called microglia. If it is in the, if it is in the spleen, then it is called red pulp. There is red pulp in the spleen, red pulp macrophage. That will be the red pulp macrophage. If it is in alveoli, like uh, lungs, alveoli. In lungs, if it is in lungs, then it is called alveolar macrophage. So this is all important things, and if it is in the skin, then it can form Langerhans. Okay, Langerhans. So these are all the tissue resident. What tissue resident means? Resident school, you know, na? Resident school where the student will be live. So this, during the embryonic development, they stopped. They stayed here. Like you, some students are staying at the home of their grandmothers. Okay, like we can say nani, grandmother's place, mother's mother. So that is called resident. From the birth they started living there only. Okay, like that. So that is called tissue resident. So from the birth only, this macrophage started living in the skin, then it is called Langerhans. It is started living in the liver, then it is called Kupffer cell. So like that, we have different type of, what we have different type of macrophage, tissue resident macrophage. So mast cell we have to read. After this mast cell, we will conclude this topic. Tomorrow we will read about what? Tomorrow we will read about T cell, B cells, and other cells, and we will read how it will form as an antigen. Okay. The next topic is what? Another, which number it is? Something monocytes. fourth number. Okay, here only I will tell you about the dendritic cells. Okay, so dendritic cells are come from the myeloid, or it can come from the lymphoid origin. Okay, cell can arise from both myeloid and lymphoid origin. Okay, it will express MHC class 2 molecule. Okay, and it will present MSC express MSC class 2 molecule, and it will go to bind to the T helper cells. Okay, so this is a like professional. It will called as a professional antigen presenting cell. It is called antigen presenting cell. Okay, so uh, it is all about what dendritic cells. Okay, so dendritic cells will be looks like this. So MSC molecule we will discuss after completion of this topic. Okay. The next is what? The fifth number is what? Mast cell. Okay. Fifth number is what? Mast cell. It is also granulated, but no color granules. Okay, color granule. If you put the dye, no color will go. No color granules. So it is called as a. It is considered under the. It is considered under the what? A granules me. Oh, sorry. So the mast cell is what? Mast cell is pluripotent in nature. Okay, these are pluripotent means it will directly derive from okay pluripotent stem cells. It will directly derive from pluripotent stem cell. Important thing is that mast cell is the similar like a. It is similar. It is similar like a. It is similar like what basophil. Okay, similar like basophil. Okay, why I am saying similar? Because the mast cell is also producing antigen response, antigenic response, and it will also produce histamine. Okay, so this is all about the monocytes. Sorry, it is all about uh, mast cell, and now I am going to conclude it. You just go and revise this from the videos. Hope so you understood this topic. If you have any doubt, you can ask me directly here. Okay, thanks for watching. Oh shit.